Hello, everybody, and welcome to our third and final session for the Mini Medical School. This module is brought to you by the Center for Genome Engineering, the promise and power of genome engineering. My name is Steph Kennelly, and I am the program manager at the center. And today we are so excited to have you here today as we're talking about cancer. On a personal note, I myself am a cancer survivor, and the stories that you hear today are incredibly important to me, and I hope that they will resonate with you as well. There's probably a chance that you've been touched by cancer in your life, so we're just so thankful that you took time out of your day to come and hear these stories today. Just as a reminder, some program tips. This will be a moderated discussion followed by a question and answer session at the end. You can submit your questions to our panelists at any time using the Q&A box that you can find at the bottom of the Zoom screen. And note that the session is recorded and will be available starting tomorrow on the Office of Academic and Clinical Affairs YouTube channel. And we will share that link in a follow-up email that we will send after the session. So today we are going to be talking to two scientists and a nurse about engineering immune cells to fight cancer and how metastasis works in the body and what we are doing to address it. So first up, our first panelist is Dr. Eric Rarman, and he joined the faculty in February 2024 and is based at the Hormel Institute in Austin, Minnesota. If you have not been down to Austin, Minnesota, they have a fantastic campus there at the University of Minnesota, and they do amazing research, especially in the cancer field. So today, Dr. Rarman is going to discuss why metastatic cancer is so challenging to research, and can you share what your team is doing to address these challenges?
Thanks, Steph. Yeah, I'm very excited to be here today, and thank you very much for the invitation. I'll just share my screen here with you all. All right. Well, yeah, I'm Eric. Yep, and we're down in Austin, so come on down anytime. We've also got a phenomenal spam museum down here. This is where the land where Hormel Foods resides and where spam is made. So come on down and visit. Today we're going to talk a little bit first about rethinking metastasis as a regenerative misfire, and I'll get into more detail about what that means. But first off, I don't have any conflicts or anything to disclose, but I wanted to spend the first bit of time talking about, well, what is metastasis? And why are we concerned about metastasis here? So metastasis really is the primary cause of cancer-related deaths. There are roughly 693,000 people in the US living with metastatic cancer, and those are across the top six most common cancers: lung, breast, colorectal, prostate, bladder, melanoma. And really, when we think about metastasis, we often think of only the primary tumor where it starts. But the vast majority of cancer-related deaths, nearly 9 out of 10 in some cases, are due to how, not the original tumor, but how the tumor spreads to other parts of the body and how that spread compromises the function of our vital organs. And despite that being the number one cause of cancer-related death, most of our treatments, models, and even research funding are really still focused on the primary tumor and not necessarily on metastasis. And so this mismatch is what really my lab and many others are trying to change. I'll talk more about that towards the end there. Okay. So what is metastasis? Metastasis really is the process of how cells spread in the body. Metastasis itself is not good or bad, but it's really what is the cell type being moved in the body. And if cells coming from tumors are being moved around, then we consider metastasis as cancer. Now, metastasis really is just a change in place. And here's an example of the different stages of metastasis. There's at least 6 major stages, but it's a very complicated process. And in this context, we think a lot about the primary tumor, but really tumor cells can leave their primary tumor and they can invade into surrounding tissues, such as their vasculature or the lymphatics, in order to spread throughout the body. As they spread through the body, they will land at some place in the body, and they'll be able to exit through the circulation to distant tissue. Let's say if there's a mammary tissue, a mammary breast cancer, it could disseminate into the lungs, and then in the lungs, when the tissues or the cells get there, they're called disseminated tumor cells. And they can either do one of three things. They can survive and grow out. They can sit idly and dormant, means they go to slumber and then they hide from the immune system for later reawakening, or they can be wiped out by the immune system and die. And if they grow out, then we have what we're known as metastatic disease or seeded metastasis. And so, as you can see, there's lots of different cell types in here. So, metastasis isn't really just isolated to the tumor cells. There are cooperating cells such as fibroblasts, which are support cells for our tissues, normal cells that are co-opted by the tumor in order to allow for cells to spread through the body. There's the endothelial cells that line our vasculature, which they themselves can change to allow for cells to infiltrate, to enter and exit. There are even immune cells that we've heard about, and I know you hear about more later from the others, that are there to fight the cancer, but some of them there are actually help the cancer to spread. In this example, we have different types of T cells called T regulatory cells that can help mask tumor cells at their primary site. We have other types like neutrophils that can create like a cargo or chariot to help carry tumor cells from the primary site into the blood and create a barrier to help them survive this harsh circulation. There are platelets that create armor. And even at distant sites, you have different reactivation of our immune cells that can create these traps or these nets that can pull out and allow for tumor cells to expand. And so this whole process is coordinated not just by tumor cells, but by many cell types. But one thing that does happen is that once metastasis has been established, the current treatments frequently fail to provide durable responses. Now, how do cancer cells know where to spread? That's been a big question for over centuries. And it goes back to a theory called the seed in the soil by Stephen Paget, when he claimed that when a plant goes to seed, its seeds are carried in all directions, but they can only live and grow if they fall on congenial soil. So some of the aspects that help or cues that direct when tumor cells, when they grow out to spread, are informed by their own genetics, so what sort of mutations they may have, or their different state. And I'll talk a little bit about that later, as whether the tumor cell is a cell that has the ability to be kind of like a stem cell. We call them like a cancer stem cell. So the tumor genetics and phenotype. What is the local tissue anatomy like where it's growing? Is it very restrictive? Is there a lot of inability, physical constraints from the cells leaving and maneuvering? We call that like extracellular matrix, which is support for our normal healthy cells. Sometimes that can get in the way of tumor cells spreading. Sometimes it can facilitate it. Access to lymphatics, vasculature, other signals from other cell types, like I was mentioning, that are pro-tumorigenic and pro-metastatic, like some immune cell compartments, some fibroblasts. So there's lots of cues and directions that allow for cells to spread, and how they spread too is different. And it's really how they manifest, I think. And it's about the abilities of the cells to survive. It's a very harsh process. At a per cell level, the process of metastasis is very inefficient because it requires lots of energy for the cells to change shape, move through the body, survive in new environments, go to distant tissues. So, for example, if a lung cancer were to spread and go to the brain, it's a very different environment for those cells to survive, so they need to adapt. So at a per cell level, it's very inefficient, and there's a lot of clearance by the immune system, which is able to get rid of the vast majority of these events that happen. But on very rare occasions, there is success. And we can see that different types of cancers, lung, breast, colon, and prostate, can present differently about where they go to and where they survive and thrive versus where they may sit dormant. So we think that there's specific cues for cells to go to various organs, but it's more as we develop our technologies more and more, it's more that metastasis has happened throughout the body and that it's just more successful in certain organs. And we're trying to understand why some of these cancers spread more successfully to other organs while other ones stay quiet. So I won't talk too much in great detail about our current treatments. So treatment for metastatic cancer is really determined by a combination of the tumor biology, the type of disease, the spread of the disease, and patient factors too, such as can patient tolerate therapies, what therapies are available to them. These include a gambit of different therapies, and you'll hear more about immunotherapy later, so I won't go into detail, but these are exemplars of the types of therapies that have happened. And there has been success in metastatic cancer treatment in the context, especially in metastatic melanoma, which is heavily driven by a lot of mutational burden, but that's not true for all types of solid cancers. So why do these treatments fail? So as I mentioned early on, the vast majority of efforts and research have focused on the primary tumor. And we know now that the metastatic tumor does not reflect the primary tumor. They may have shades of what they look like originally, but they have undergone this basic stress and adaptation through all those different processes of leaving the primary site, surviving in circulation, evading clearance by the immune cell. They've been sculpted to be very efficient and to survive in their new environments. So they only look like a shade of what really the primary tumor is. There's also other aspects that can have more complex tumor microenvironments or interactions with different types of cells. Organ sites of metastasis and treatment delivery. So treatment to the brain is going to be different from treatment to the lung, which is spread to the liver, for example. There's also oftentimes the cells have abilities that make them more therapy resistant and adaptive to the tumor evolution process. And what we've known for the longest time is thought that there's a silver bullet, that there's a single mutation that causes metastasis or cancer. But what we've found as a field, the royal we, that vast sequencing efforts have determined that there's really no single metastasis causing mutation. And it's really not just a genetic issue, but a systems level problem. And what I mean by that is it's not just about the tumor cells and the mutations they have. They have the ability of adaptation to be like a stem-like cell. So we have to fight that in a different context. They have different sort of armors with the surrounding cells around them that are providing a pro-supportive and surviving environment. So that's what we're talking about when it's a systems level problem. So oftentimes when we talk about metastasis, people ask, well, can you just stop it before it starts? And really the key aspects of what's been most impactful in cancer prevention or in treatment is earlier intervention and detection early on, such as more advancements in mammograms, more and more colonoscopies, skin checks more regularly, having healthy habits, which generally keeps stress and on the body low. Stress creates an environment that is pro-cancer spreading. We've starting to see many more studies coming out demonstrating this, that altering the immune system by creating a stressed status is more pro-cancer spread. There's also new frontiers now in prevention. As I mentioned, when cancer cells spread and they land in distant organs, let's say if it's a gastric tumor going to the liver, cells can either grow out and form vast metastatic tumors. They can sit idling dormant or in this dormant state. So there are many labs now working at like, well, if the process has already happened and they've spread and we can't stop before it spreads, can we keep them at a slumber? And if we keep them in slumber, can we then look at a state of then eliminating them while they're asleep before they grow out and compromise the function of our organs? So now I'm just going to shift gears a little bit. That's a bit of a backdrop on metastasis. I'm going to tell you a little bit about what my lab does and how we're upending the idea of metastasis a bit more and putting in the context of a normal process that is corrupted by tumor cells. So we've thought about cancer and development, that cancer for a long time has been sold as something that is special or unique. Well, cancer really only uses processes that already exist to a cell. They unlock a toolbox that's locked to them, but this exists during these processes exist during some stage of development, whether it's post-conception, during fetal development, post-birth, or during puberty, or even in aging. These processes exist and cancer corrupts them at inappropriate times. So if the process of cancer is not inventing anything new, then the process of how they're spreading the body, that must also be a normal process that has been hijacked. And when we think about it, let's use this example of these homes here, where each home is a unique organ. And we'll have the lung on the left, the liver in the middle, and we'll have the kidney on the right and white. And what we always thought about, these bricks are representing a cell type called an epithelial cell, which for many years we thought to be rather stagnant. They are workhorses of our organs. They provide structure and order and function to our organs, and we think that they reside there. Well, through many years of work during my postdoc, we realized that actually there's a lot more of fluid exchange between our organs than we've appreciated. There is mosaicism that happens where we can have example of lung cells going to liver and contributing to the liver, but not actually forming tumors. Same with other types of cells, such as kidney going to the lung. So really, metastasis is not a phenomenon of cancer, really. It's really a process, a normal physiological process hijacked by tumor cells. So just to summarize very high level, what I've spent a good chunk of my life working on is that we've identified a specific type of protein, an ion channel. It's called a sodium latent channel or NALCAN. It allows for sodium to flow into the cell. It regulates the cell's bioelectric properties or its membrane potential, or basically the resting state of the cell. And what we've identified that this channel gene, when it's highly mutated in several types of human cancers, and that when we model this in genetically engineered mouse models, so this is what we use a lot in my lab, are genetically engineered mouse models to mimic human conditions, that when we delete this channel, either one or two copies, so in DNA we have two copies of each gene, that we will delete one or two copies for heterozygous or knockout state, that when an animal already has a tumor and we layer this on top of that, it causes the tumor cells to spread into the vasculature and increase the presence of seeded metastases. So this channel seems to be operating as a way to promote metastatic dissemination. Now, my training has been in developmental biology, and so I want to know, well, what does this channel do? So when we just looked at this channel now, we created genetic deletions in these organs, same organs, in this case, we use the stomach as an example, is that deletion of this channel alone didn't cause cancer. It's not a cancer-causing mutation, but what we did find is that cells from the stomach would actually actively migrate into the bloodstream and go to distant organs and seed in these distant organs. But unlike their tumor counterpart, when they seeded in those distant organs, they incorporated into the normal architecture of this organ. So we have gastric epithelial stem cells going into the blood, going to the kidney, and making kidney structures, right? And although this happens at a very rare frequency, it does occur, and that this channel is a gatekeeper for this, and that we can tune this up or tune it down if we alter the channel. So we can think about metastasis now in two aspects of like, how do we dial down bad metastasis in the context of cancer to prevent further spreading of tumor cells, or could we ramp it up in the context of regenerative medicine? Could we actually help our organs repair themselves better by manipulating the expression or function of this ion channel? Now, since my lab started, I moved back to Minnesota in February of 2024, and we've really been asking this question and investigating metastasis as our model system, cancer metastasis, as a rewiring of normal physiology. We use a variety of different transgenic mouse models. We use organoid systems, which are basically mini organs that we can grow in a dish from either human tissues and or from our mouse tissues. And we can use approaches like CRISPR that you've heard about, and you'll hear a little bit more about that, to genetically modify these organoids to basically run metastasis in a dish. We also use a variety of different human cell lines. And really at the crux of this is understanding this channel, this protein that sits at a cell membrane, how it controls electrical activity of a cell, and how that determines cell movement, cell communication, and homeostasis, which is our everyday maintenance of our organs. And that disruption of this process can lead to disease. In this example, we'll talk specifically about cancer metastasis, but we think that other diseases are like metastasis, like fibrosing disorders. So we ask major questions. How do we stop cancer metastasis? Can we improve the targeted treatments? And then in a regenerative medicine aspect, can we promote better health of our tissues? Now, as I mentioned, the process of metastasis we think is a normal physiology aspect. Here again is the example that we've had. I'm going to give you some very high-level examples of experiments that are running in my lab. We have, for example, we're looking what's happening, what is this ion channel doing at the actual tumor cells? And one of our focuses is looking at the sensory antenna of a cell. So many of our cells have this little projection here called the primary cilia that works like a sensory antenna that brings input for what's happening from the surroundings. It can take in information about availability of nutrients. It can talk about pH. It's a great sensor that has lots of signaling hubs shown in this cartoon here. What we've identified is that NALCAN sits at the base of this primary cilia and something called a basal body here. And when we take mammary epithelial cells and we get rid of NALCAN, we see that we lose primary cilia. And what we know about primary cilia is that when it's gone, cells become or adapt to become more cancerous, meaning they can enhance proliferation, but they can also enhance migration and invasion. So we think that this is one aspect of how this ion channel is orchestrating the ability of cells to leave is through this sensory antenna. The second is that we're looking at the endothelial cells. We've realized through a variety of our experiments that this ion channel is also expressed in these cell types and that they are involved in blood vessel. We think that they're involved in blood vessel function. Now, endothelial cells their barrier can fall apart a little bit during tumor formation. They can become leaky, and that promotes tumor cells for spread in the body. We found that if we run an in vitro assay where we plate endothelial cells in a dish, they form these beautiful matrices after 1 day, that if we block this channel, we block the ability of them forming this. So now we're running the in vivo mouse experiments. And lastly, we're looking at what's happening at the seed of metastatic site. We identified that these fibroblast cells, which are support cells in our tissues, express the sodium latent channel, and that we found that these cells can convert to something called cancer-associated fibroblasts that become pro-tumorigenic to allow tumor cells to grow out. What we've identified is that this process of this conversion from these fibroblastic cells to cancer fibroblasts is blocked when Nalcan suppressed. So it has a role in this conversion process. So we're looking at this large systems level approach. Now, as I mentioned, metastasis is highly complex. We've got our tumor cell, which has initial mutations, progression, epigenetic changes, which are huge for metastasis. We need to start thinking about the soil as well, the local microenvironments, the stroma, inflammation, hypoxia. These all contribute to that. Our climate, which is what's happening in the host, our genetic backgrounds, what predisposes us, not necessarily mutations, but other types of alleles that may make us predisposed to cancer metastasis, even our immunity, and also therapy. We're realizing now that some therapies on the early stages of being really advantageous of treating that primary tumor can inadvertently lead to some sculpting and clonal expansion to make things a little worse. And so we're starting to look at that as well. And then altogether, all these things contribute to metastasis. So to really address this, one lab can't do this. So when I moved back to Minnesota, I put a few heads together and we developed this Minnesota Metastasis Research Network. And it's really a combination of efforts between the labs here at the Hormel Institute, which I oversee, the Mayo Clinic by Gina Razidlow, and then Kaylee Schwartfeger at the University of Minnesota. And together, these three institutes were coming together to find new strategies to treat metastasis. And we're looking at the early stages to the mid stages to the end stages, and even how do we translate these into efforts that are being done for those in clinical trials. So this is MinMet for short. But yeah, so we're over 25 labs now, and we're sharing these expertise in order to tackle metastasis faster. And one example of this is that we've come together to look at the effects of stress responsive programming on the dormancy and metastatic escape of colorectal cancer. In this context, it brings labs across the University of Minnesota, Twin Cities, Mayo Clinic, and the Hormel Institute to use genetically engineering models of mice as well as organoids with approaches like CRISPR in order to understand how does stress, whether it's inflammatory bowel disease, the food we eat, or even chemotherapy regimens, impact the spread of cancer. So, with that, I thank you for your time. I hope you learned a little bit about cancer metastasis, and I'll take any questions towards the end during our question and answer session. So, thank you very much.
Thank you so much, Eric. That was really a helpful overview. I especially like the visual of the brick house and how um, the cells play a role there. As a reminder to everybody, you can continue to submit your questions down in the Q&A box, and at the end, we will answer once all the presentations are complete. Now I'd like to introduce you to Dr. Brandon Moriarty. He is the co-director of the Center for Genome Engineering. He runs a basic translational research laboratory working to develop novel cellular therapeutics for gene therapy and cancer immunotherapy with the goal of translating new therapeutics to the clinic. And I'm thrilled for everybody to hear more about the promise of TIL therapy, which he's going to be discussing. So Brandon, can you please share more about this exciting research and the promise of the upcoming clinical trial?
Absolutely. Yeah. Thanks for the invitation to be here and excited to tell you about some of the work that we've been doing at the center and in my laboratory over the last few years. So the title is Treating Advanced Cancers with Engineered Immune Cells. No disclosures for me either. So first of all, I can inform you that we are genome engineers. And so what is this? So we actually change the DNA code of cells to change their ability to function and operate. You can think about this like an automotive engineer changes the blueprint design of a car to make it better, faster, more fuel efficient. So, for those of you that didn't know, the DNA in every cell of your body is like a blueprint or an informational book that tells the cell how to build itself and make copies of itself. And so, by changing that code or blueprint, we can give them new functions. And so, how do we actually do genome engineering? Well, one of the exciting things is that two of the technologies to modify the genetic code of cells were developed here at the University of Minnesota. So, one was Dr. Perry Hackett, who invented what are called DNA transposons. That's where we can take synthetic DNA fragments that we generate in the laboratory and we can insert those into the genome of a cell. And within that DNA code are instructions to build new components for the cell to change their function. Secondarily, a precursor technology to CRISPR-Cas9 called TALENs was developed by Dr. Dan Voitas. And what this technology allows us to do is actually to change the genetic code of the existing DNA. And so your DNA is made up of A's, C's, G's, and T's like code. And so we can go in and systematically change those to change the phenotype or function of the cells for, in our case, therapeutic purposes. And so we focus primarily on cancer immunotherapy and using genome engineered immune cells. So some of you may know that your immune system, that's made up of many different cell types, is defending you from bacteria, viruses, and cancer. In fact, it's your best defense against developing cancer. And only when your immune system fails to detect developing cancer is it allowed to grow and thrive to a clinically detectable form of cancer. And so it seems intuitive to us and many in the field that if your immune system's evolved to have this job, then we should deploy them for this purpose for therapeutics. And in this diagram, what you can see is that our body has many different types of immune cells that have lots of different functions. Some of them can be used in a what we call autologous or personalized fashion, where the cells that we use for therapy have to actually come from the patient. And so these are a very popular cell type because they can be very effective for cancer immunotherapy, and these are called T cells. And then there's another set of immune cells that we call off the shelf or allogeneic. And that's where we can actually take these cells from a healthy donor and then engineer them and use those to treat a cancer patient's existing cancer. But it's still to this date, even though there's lots of activity in these different cell types, it's very unclear which cell type or approach will be the, silver bullet, the cure for cancer. And so, in one iteration of this, using that very personalized approach, is where the field's made huge strides in treating leukemias. And so, this is where we simply take blood from the cancer patient, we isolate those T cells that I just mentioned. They are engineered with a special receptor that I'll tell you about in a moment. They're then re-expanded to billions in number and reinfused into the patient. And this really is a form of cancer immunotherapy that has cured thousands and thousands of people with leukemia. And so the most prominent version of this is called CD19 CAR T therapy. And so this is where we take the patient's T cells, we insert a sequence of DNA that encodes for a new receptor called a chimeric antigen receptor. So you can think about this like a metal detector. So we're giving the T cells a new tool, a metal detector in this example, where it can actually identify the cancer cells and allows them to kill the cancer cells specifically. And this has had a phenomenal deployment in therapeutics for leukemia, as I mentioned. And so over here on the right is just a graph of leukemia event-free survival. And so prior to the development of CAR T, the survival rate for these leukemias was only 10%. And now with CAR therapy, we're seeing durable cures of almost 80% in patients. Unfortunately, these types of therapies have not done as well in solid tumor cancers, and we and many other people are working on this. But in our laboratory, we've decided to focus, in addition to another form of cancer immunotherapy called tumor infiltrating lymphocyte immunotherapy. And so this is a technology or approach that was largely developed by Dr. Steve Rosenberg at the National Institutes of Health Surgery branch. And so here he knew that most tumors in his patients were full of immune cells. So the blue cells are the cancer cells, and the little different colored dots here are represented as T cells. And he hypothesized that those cells were there for a reason, that they were trying to fight off the cancer, but they were losing the battle. And so what he would do is he would take a small piece of tumor out of the patient. He'd cut up the tumor into little fragments and he'd put each of them in a little well of a dish in his laboratory. And he'd give them lots of nutrients that they really like to grow and thrive on. And so he'd grow these T cells up in these dishes. And then he'd actually test them against the tumors that he took out of the patient, and he would find ones that indeed in a dish could kill or be activated by that tumor. And then he would only grow those cells up to billions in number and reinfuse them into patients. And for the sake of time, I won't give a lot of examples, but this has cured people of highly metastatic solid tumor cancers, particularly in melanoma and lung cancer that have high mutation rates. And so it's much easier for T cells to see cancer types with high mutational rates like melanoma and lung cancer. Um, and so, really, we've been focusing on this in our translational genome engineering lab to take this to the clinic, but not only this procedure that I just described, but now deploying genome engineering to change the genetic code of those T cells or those TIL to be more effective at fighting solid tumor cancers. And so, just a little background on how we take things in the laboratory from kind of concept to clinic. And we really see this as three stages. The first stage is we have all these different immune cells we're really interested in this case, alpha beta T cells. But first, we have to optimize our ability to change their genetic code, typically using things like CRISPR-Cas9. And so in some cases, we just want to be able to turn genes off. And sometimes we want to bring in new genes to give them new functions. And then we also have to be able to grow them to very large numbers, millions and billions in quantity, because we know from previous studies that you need lots of immune cells for the therapy to be really effective. And so once we have our ability to change the genetic code of our given immune cell worked out, then we start partnering with clinicians and specific indications of cancer, and we devise an approach or a plan of which genetic edits we want to change, what new receptors might we want these cells to express. And then we optimize our ability to change the genetic code of that immune cell in the exact way that we intended. And we perform preclinical testing for both efficacy and safety. And so we'll put them into animal models that have tumors, and we'll see, do they eradicate the tumors? Do they harm the animals? We do assays where we put them into a dish in the laboratory with normal human cells and ensure that they don't kill normal cells, things of that nature. And then once we have something we think is very exciting, we want to take to the clinic, we work with our GMP facility or good manufacturing facility. Luckily, we have one of these in St. Paul. And only when cells are made in this building, under all of their regulatory and paperwork filing, can they actually be put into people. And so we take our genome engineering approach and we teach them how to do it in their facility. And then they scale it up and show that they can reproducibly do this genome engineering. And then we take the data from all three stages and we file what's called an investigational new drug application with the FDA. And essentially, they'll come back to us and say, we need more safety data, or they might say, this is satisfying us. And for this given indication, we're giving you the green light to start the phase 1 clinical trial. And so that's the process of how we take something kind of from concept to clinic for cell therapies. And so over the last 10 years, we've actually been able to launch 6 clinical trials. Doing this approach for various cancers. And today I'll tell you kind of one vignette of the clinical trials that we've been able to get into the clinic. As you can see in this diagram, we have a lot of different projects in the laboratory that are marching towards the clinic. Some of them are led by the U of M, which is the story I'll tell you about today. Some of them are actually out licensed to existing companies where they carry out the clinical trials. But just to kind of give you an example that there's many of these different therapies coming down the pipeline, and they can be executed either here at the U of M or by company collaborators. And so, as I mentioned, the vignette today will be on the first in human phase 1/2 trial using genetically engineered TILs to treat highly metastatic GI cancers. And in this case, GI cancers is kind of a catch-all, including colorectal, pancreatic, cholangiocarcinoma, anything of the GI tract. And so this all started from a collaboration that we had with the NIH where they had identified a gene called CISH. So this is a gene in everyone's genome. And they found that this has a special function in regulating T cell activation or T cell function. And so they found out when T cells are activated, for instance, when they find a virus or when they find a tumor cell, the cell turns on this gene called CISH to help regulate or dampen down the T cell so it doesn't get hyperactivated and induce autoimmunity. But what they found is that they made a mouse model where they actually removed the CISH gene, so these mice no longer had this. And then they harvested tumor infiltrating lymphocytes from this mouse that came from a melanoma tumor, and then they treated those mice. And what they found is that when they would give the TIL from wild type mice, the tumors would grow in size. So this is tumor size. You can see they're just getting really large, even with wild type TIL. But when they used the TIL that were replete of CISH, they saw complete responses in all the animals and 100% survival. And importantly, they did not observe autoimmunity in this mouse that lacked CISH. And so, based on all of this, we decided, well, this might be a good gene to turn off in TIL and treat patients with metastatic GI cancer. And so, what this slide is depicting, we won't go too deep into the details, but just the highlights of this is the steps we had to take to go from concept to clinic, that preclinical work, stage 1 to 3 that I just walked you through. And this was published in 2022. Well, first, what we showed is that indeed in human TIL, when we activated them, the CISH gene did turn on. So it was doing what it does in the mouse. So that was a good, step in the right direction that, this is recapitulating what we saw in mouse. We then optimized our ability to turn that gene off using CRISPR-Cas9 technology. And the punchline here is that we could treat TIL cells, and 95% of them we could turn off the CISH protein or the CISH gene. So these are control where you can see CISH is there and then CISH is gone. And we also showed that this editing was very specific and only turned off CISH and not any other genes in the genome. We then further showed that T cells from peripheral blood, if we turn off their CISH gene and then activating them, they would have higher functionality. They were better at doing their job and killing cancer cells. And then finally, we took TIL from patients that Rosenberg had done surgery on. And indeed, we showed when we turn off the CISH gene in TIL, they're better at doing their job. They're more responsive to cancer cells. They're better at killing them. And so that was really exciting. So then we went to stage 3 and showed our GMP facility how to do the manufacturing. They showed they could do this several times with success. And then we filed our IND and got approval for our first in human clinical trial using engineered TIL. And so this is a synopsis of the results of this study, and this was done in collaboration with Dr. Emil Liu here at the U of M, where essentially these were highly metastatic end-stage GI cancer patients. They went through 7 to 12 previous trials or therapies, and this was basically their last hurrah. This was going to be their last therapy before they expired. And so we enrolled 22 patients. Some of them became ineligible because their cancer progressed too quickly, or some of them we were unable to isolate TIL from their tumors. But in the end, 12 patients were treated with our engineered TIL therapy. And excitingly, all of these patients, their tumors and metastases were rapidly growing, but with the treatment, about half of them had stable disease at 1 month, so everything stopped growing. A third of them had stable disease at 2 months, everything stopped growing. But unfortunately, for 11 of 12 of these patients, their tumors then started growing again and they all succumbed to their disease. However, we did have one patient, patient 12. She had highly metastatic colorectal cancer. We identified TIL fragments that were reactive against her tumor. That's what these blue dots are depicting. And so we decided to make her a clinical product where we turned off the cyst gene very effectively. And then we gave her one single dose of this product. And what we found by CT scans over time is that over the next few months, her tumor started shrinking, her metastatic lesions started disappearing, and by about 7 months, she was completely cancer free. So here's the CT scan before showing metastases in the primary tumor, and then at about 1 year, they're completely gone. And so this patient's now been cancer free for 2.5 years and is showing, this therapy can cure these incurable, highly metastatic cancers. But the therapy is not accessible enough right now. It's too complex, and it needs to be enhanced further. So instead of one of 12 cures, maybe we can get, 6 of 12 cures. And so that's what we're doing now is developing the next generation of engineered till by first avoiding some of the complex selection process, like those till fragments where we have to figure out which ones to use. And so we've overcome that. And we've also enhanced our genome editing. So we're doing more than just turning off CISH. And we've gotten the engineering pipeline down from 3 months to make the product to 20 to 24 days. And in our previous trial, it was $500,000 a patient, and we think we can get it now down to about $150,000. And so, with this, to enable these next steps in the next clinical trial, we initiated what we call the Minnesota TIL Alliance. It's a conglomerate of researchers and clinicians here at the U of M and Mayo Clinic, and a fundraiser called Until It's Cured to raise funds for the next clinical trial. And so these are the clinicians we've partnered with at the U of M and Mayo Clinic, and you can see all of them have different cancer types of expertise. And our next trial will actually be treating all of these different types of cancers in a basket trial. And so here's an overview of the Until It's Cured mission. So we're looking to raise $5 million by Q1 of 2026. About 1.5 million of that is for research to reduce the timeline cost and further enhance TIL function. The rest of it will actually be to run a clinical trial slated to start in Q1 of 2022, where we'll treat 20 to 30 patients with the end stage cancers of those various types on the previous slide. And then, if successful, of course, we will move this into commercialization and larger phase 2 clinical trials. And I'm excited to say that we do have over $1 million committed just starting this fundraiser in July. Still a long ways to go, but I think we'll get there. And with that, just want to thank everyone. You can imagine something this complex takes a village. So this is just an acknowledgement to all the people involved in the funding. And with that, I will say thanks and take any questions that you guys have at the end of this session.
Thank you so much, Brandon. I want to make sure that people know we are hosting a special event on November 13th, and we can send information in our follow-up email. But this event will be showcasing the TIL labs and be part of a fundraiser with a local partner, Vranicure, Vranicure, if you want to check them out. So if you're interested in learning more about supporting this research, we would love to have you at our event on that day. So next, we are bringing in Sarah Crane. Sarah is a clinical trial recruitment coordinator for the Developmental Therapeutics Initiative and the Masonic Cancer Center. She has a strong background in both direct patient care and clinical research. Sarah has a real-life perspective on bridging the gap between cutting-edge medical discoveries and the people who need them. So Sarah, welcome, and can you please discuss the role of nursing in cell therapy clinical trials?
Yeah, hi, Steph. Thank you so much for that introduction, and thank you, everybody, for attending this session tonight. It's super exciting to be here, and I really appreciate the mini medical school considering the voice of a nurse, since any of you that are about to go to medical school will soon find out that nurses will definitely tell you lots of opinions and things that they need to have a voice about. So we're very good at at mentoring, and so I appreciate that that I can be here to talk with you tonight. So I am a clinical research nurse, and like Steph said, I was actually a nurse on the trial that Dr. Moriarty was just talking about with um, the CRISPR-TIL study. And so that's kind of where I come to be here. And I just think it's a great idea to talk about the role of the nurse. And you know, we had the one patient that has been mentioned, Emma, that had a complete response, but there were several others that had that stable disease, and all of them were so brave and so wonderful. It was such an honor to take care of them and to, you know, advocate for them. So my first slide, I'm already critiquing myself. I feel like it probably should have been more of a Venn diagram, but showing some overlap here. But the way I think about it, and the clinical research nurse is called a clinical research coordinator. So we actually call them CRCRNs. And the patient and the study are kind of our two worlds that we're that we're in charge of in this job. And so on the patient side, you can imagine if this is your third session and you've been to all three sessions, you can see how education is very, very important. Being able to break some of this language down in a way that is understandable is really important. And I actually even have some parents that are getting these therapies, you know, different TIL therapies, different cellular therapies that say, you know, can you give me some idea of how to talk about this with my kids? And then I'll do that. And they said, well, I also told my kid's teacher that this is what's happening, and the teacher is going to use this as a lesson. And then, you know, then it goes to the church, and then it goes into the community. And so this education piece is not just for the patient. It is really something that they can use to empower themselves to speak about these therapies and it's really complex issues. So that's the education, the part, but also there's a time for seriousness too. You know, the consent form that I have to go over, make sure that they understand every single word. And it is confusing, but that is the time to ask questions. And if you have to absolutely understand what is about to happen, because like Brandon said, these people are volunteering, but they are very sick. They are, you know, stage 4, you know, and they have to understand the risks and the benefits and have time to weigh that out themselves, you know. Also, another part of this individualized care is the nurse also helps if there's problems with like caregiving or lodging or you know, car rides. The caregiver piece is so huge because sometimes the caregiver might have medical issues of their own that they're also dealing with. And so a nurse really has to take this kind of global approach to their care and make sure that all of these pieces are kind of in place and can kind of work well with one another in order to make it as seamless as possible for the patient to volunteer to do something like this, because they're really helping us out by volunteering. And then the other side of it is the study. Of course, we have to be very, very particular, about what we do and who we enroll. There's the protocol, which is kind of every study has a protocol. That's kind of the instruction manual for what you should be doing. And so a nurse's role in that aspect is when, for example, with that CRISPR-TIL study, when the patient gets their TILs infusion, it's our job to watch like a hawk, and also to ask questions. How are you feeling? How are you feeling? They're going to get really tired of hearing that. If they get a fever, when did the fever start? When did it end? What did we do? How did we treat it? Did they get a fever the next day? We're really tracking every little thing. If there's even a little bit of a rash or anything, we need to know absolutely everything that happens. And another part of the study too is that we're overseen by regulatory, lots of regulatory boards, the FDA, the University of Minnesota. There's all of these people that watch over to make sure that we're safe, because that's obviously number one, is we have to be safe. But just the sponsor, the person who sponsors the study, or the company that sponsors the study, are they doing what they need to do? Are we doing what we need to do? And so there's a lot of communication that the nurse does with the company that runs the trial. So we meet in meetings, on the CRISPR-TIL trial. I was in meetings sometimes with Brandon and Bo Weber and Dr. Liu, and we were all kind of talking through the patients together. What should we do, and how is everybody doing, and things like that. So it's very interesting. So those are kind of the big responsibilities. And then the data that's collected now, that goes into like a big database, and then thank goodness the nurse is not in charge of analyzing that data or putting it all together and making sense out of any of it. Thank goodness. We are just there to kind of collect information, but we do use our nursing skills to develop plans, care plans for the patients and their symptoms. So also working together with pharmacists, social workers, nurse coordinators, the doctors, of course, and other nurses. So everybody gets involved. It's a big team collaboration. Speaking of the education piece, I would like to just show you a couple examples of how I explain some of this complex things that Bo and Brandon and other most of my clinical trials, they either are cellular therapies like what Brandon's been talking about, or they are immunotherapies. And so I'm going to explain the difference between the two. Did I choose this to talk about this so that I could put a hippo in my slide deck? Yes. I always like to say that immunotherapy, I tell people to kind of try and picture a hippopotamus and how hippos have those birds that live or that, sit on its back. And for immunotherapy, you kind of have to think about chemotherapy, which is some of the first kind of standard of care stuff that we do. Chemotherapy, it goes into your body. And if you imagine the hippopotamus is like your cancer cell, and the birds are the things that live on the outside of your cancer cell. They are enzymes and proteins and nutrients. And cancer is evolving so quickly. Cancer is always separating, and it moves quickly and it divides quickly. And so in order for it to do that, it needs these birds to keep it clean, keep things organized, eat the bugs, get rid of all the stuff that's not supposed to be there. And that's what keeps the hippo going, is this relationship with the birds. But with chemotherapy, the hippo dies, the birds die, the grass dies, the water dies, the trees die. It's like all of a sudden you've got all of this stuff that was destroyed by the chemo. And so the immunotherapy is just for the birds. The immunotherapy is designed to have a block or a lock or a block or some type of system that blocks the birds from landing on the hippo. Sometimes it blocks the birds from eating certain bugs that it has to eat. Sometimes it blocks, different elements of this relationship. And so by doing away with the birds, you ultimately do away with the cancer itself, because it doesn't have what it needs to keep thriving and surviving. So that's kind of one of my kind of funnier examples. But a lot of people like that one. This is my imagination. And so if you find it even more confusing, I'm sorry. So the next thing I can kind of explain, even though Brandon just did a really good job, is about TILs too. When I explain the TIL trial, I would tell people about, just like Brandon said, that for some reason, there's these lymphocytes, which is kind of part of your immune system. There's lymphocytes inside of these cancer cells. But so they're the T cells, which is the T lymphocytes, they're also called T cells. The T cells, I think of tumor finding, as T cell tumor finding. They are really good at finding the cells that need to go. But some of them have a special sauce that allows them to get into the tumor, which you would think is golden, because then they can alert the immune system. But they get turned off. And Dr. Moriarty just talked about that SISH, the things that turn off that switch. There's probably lots of different things that can turn off a switch, but I like this picture of these soldiers. They're fighters. They go and find the cancer. They found the cancer. And now once they're inside, they're just kind of hanging out. They were turned off. They lose connection, and they're just kind of waiting for their next assignment. So without being able to connect, they can't call the rest of the immune system to come and help them fight. So they just kind of hang out in there. So that's why Bo and Brandon could take those. And I actually do call them Bo and Brandon when I talk about this CRISPR TIL to the patients that enrolled on the study. I would literally say, Bo and Brandon are going to take your tumor and they're going to find out who has the special sauce, how'd they get inside, who are you guys, what are you doing? And then they're going to take those and introduce them to all kinds of new T cells that are just kind of naturally floating around your body. And some of those ones that are floating around your body have the special sauce too, but they've never met each other. So we're all going to kind of grow these and CRISPR them and make them so that they're not going to turn off using the CRISPR-Cas9. And so it was really funny. Sometimes they would call me, like Brandon said, the TILs would take a couple months usually to make. And so the patients would call me and they'd say, how's Bo and Brandon doing with my cells? And I'd say, well, they're working on them. They're working on them. I said, well, tell Bo and Brandon to hurry up. And I'd get like different people kind of chiming in. Oh, how's Bo and Brandon doing? Oh, they're good. They're good. They're, working on your cells. And so I think it was kind of cool to have a name to the product, kind of like it made it more of like connected, so it was kind of funny. Um, but anyway, once they get this infusion, I put can't stop, won't stop. And literally, sometimes they can't stop. Like there's a whole thing that can happen when those cells really do go crazy and they can alert the immune system and they're like, hey, we're in here and it's freaking crazy. There's cancer all over the place. And so the immune system's like, oh, oh no. And they start to kind of run into each other and like, And so there's a way that we can kind of calm. Let's do this calmly. Let's do this, so there's different medications and stuff that we can use to kind of help organize the little chaos that can happen when it goes in there. So anyway, that was a kind of a way for me to kind of describe it. Hopefully it sounds exactly like what Brandon said, because, we're on the same wavelength here. So I just wanted to acknowledge that one of the things that Bo and Brandon and other people at the cellular engineering group, they may not understand how much they are changing the way that we deliver nursing care. You can imagine that if you have a heart problem, you go to a cardiology floor. And if you have a surgery, you go to a med surg floor. And if you have, um, you know, cancer, you go to a cancer floor. But how do you organize nursing in a way that optimizes the treatment instead of the disease? So now you're looking at, well, do we make like a cell therapy floor? Like we used to have floors that were catered to certain diseases. But with this stuff, think about it. I mean, you might have 5 different patients getting cellular therapies or immunotherapies of different kinds. One's got pancreatic cancer, one's got ovarian, one's got leukemia, one's got, and most of the cellular therapy nurses, including myself, were just originally trained in bone marrow transplant and cellular therapies. So that's kind of what we know, leukemia and lymphoma. But now here comes somebody with, uh, grade 4 or stage 4 colorectal cancer. So I just want to give a special shout out. There's going to need to be a lot of care and training and patience and all kinds of things. The nurses that are on the 5th floor at the University of Minnesota M Health Hospital are amazingly incredible. They are the ones that took care of these patients that were on the GI CRISPR-TIL study. And in order to work on this floor, the staff, I feel like there are absolute musts. You have to be curious. You have to have an all-in, gloves up, roll up the sleeves attitude. And you have to have this very wholehearted, holistic approach. Like I was talking about the community, I was talking about the kids, I was talking about these things. The nurses are involved in this. And this fast-paced innovation, this is so awesome. And the TILs and all of the stuff that's happening is so amazing. But it comes up against things like short staffing and nursing shortages and things like that. And so these nurses arrive on this floor every day, not knowing exactly what they're getting into, but they absolutely are mentors, they're learners, and I just have much, much, much respect for what they do. And so if you want to know more about clinical trials, I could probably have just a talk on that, but you can go to clinicaltrials.gov. That's always a really good resource for me. It's kind of a little complicated, but um, it really is a good um, layout of all of the different clinical trials. There's also cancer.org. That's a really good website just for anybody with cancer in general, but they also have a section that's about clinical trials. And then I also just want to say that there are AI companies now. I didn't want to mention any here because I don't know, but there are AI companies that use the clinicaltrials.gov as a database, and they will find clinical trials for you, so that you can continue caring for your loved one while somebody else is doing all the legwork of finding these clinical trials. So just some ideas there. And thank you. Thank you so much. And I really appreciate it.
Thank you, Sarah. That was fantastic. We got some feedback from Allie and Jenny, and I know I was thinking the same thing, that you were giving very caring vibes and that the perspective of a nurse is so important in these conversations, because I know that Brandon would agree that it cannot happen without you. So thank you so much for sharing, and I think it was really an important aspect of the story.
Absolutely.
Uh, yeah, so now we get to move on to the fun part. We get to get your questions. So just as a reminder, down at the bottom is the Q&A box, and you can click on that and put in your questions, and we'll get through as many as we can until 6:30. And so keep them coming. So we're going to start with Eric. Eric, you had mentioned in your talk about mechanisms that cause stress and that can help spur metastasis. Is there anything you could elaborate on that? Like, what specifically have you found in your research that causes stress?
Yeah, this is a very excellent question. Thanks for that. So this means it's a couple different things when we think about it, because stress is a systemic problem, right? It affects not just tumor cells, it affects the immune system, it affects cells that are supporting our normal healthy tissues. So one aspect definitely is cortisol. That is actually one that can help to promote metastatic spread, and its functions are, how it's acting can depend on the cell type it's acting on. So some of these systemic stressors can alter some of these pro-metastatic immune cells, like neutrophils, like I was mentioning, that make them more aggressive, or their ability to hide the tumor cells more effectively, to let them grow and fuel and actually survive when they get to the distant site. So one case, stress can alter our normal immune system to make it some of the cell types more adaptable for allowing tumor cells to grow out. In the context of the tumor cells themselves, it can impact their metabolic activity. It can create, we call it metabolic plasticity. What it really means is that the cells can adapt to use different energy sources a little easier, and survive harsh conditions. So it's stimulating how these stress molecules, when in circulation, can stimulate the actual tumor cells to allow them to adapt locally, and change part their metabolism. It can change other things about them too, including primary cilia. Stress can alter the state of the primary cilia, and stress also acts differently on tumors. It's not always bathe the whole tumor. You can create these cells, we call them resistor or persister cells. So you might be able to debulk the vast majority of a primary tumor, metastatic tumors, but some of these cells, when expressed to stress in the environment with the other cells participating, can hide away, and they can tuck away, and they might reemerge 10, 30 years later. That's what we're seeing oftentimes in breast cancer patients, that they might be cured for 10 years, and then other reemergences or reawakening of these cells that could have been spread early on are starting to emerge. And then, unfortunately, we're seeing more cases of brain metastases in patients with breast cancer, because we're getting very good at treating the other aspects of some of the solid tumors in lung and liver, but other ones are escaping. So it all is a very systems level. It's a roundabout way of saying that there's a lot of players involved that can impact this. So it's not just one player.
Great, thank you so much. Brandon, we had some questions about funding. I know we kind of plugged some of the funding philanthropy that we're doing, but amidst the changing political landscape, cancer research funding, government shutdowns, can you speak to a little bit about how does your lab get the funding to do the research, and how does the government kind of play into that?
Yeah, definitely. So the majority of academic research funding comes from the National Institutes of Health, and there's also mechanisms through the Department of Defense. The budget at NIH is around 45 billion a year, and typically the Department of Defense is around 1.6 billion. Unfortunately, the new administration thought it was a good idea to cut the Department of Defense budget by $1 billion. And that's really unfortunate because that funding goes to high risk, high reward, also cellular therapies and translational research. So that's a huge hit. Luckily, it's looking like they're not going to massively cut the NIH budget. That said, the shutdown is canceling grant reviews, so submitted grants that are waited to be reviewed by NIH, and the review sections are all canceled, and so everything's going to be massively delayed. And new awards are not being awarded because of the shutdown. Essentially, all employees at NIH are locked out of their computers, can't answer emails, can't deploy grants, can't review grants. So huge problem. The shutdown is related to that funding mechanism. So I think that answered the question, hopefully.
And how else does your lab get funding outside of the government grants?
Yeah, I mean, another great source is philanthropic organizations, like local ones would be like CCRF or nationally, the American Cancer Society. And so those usually aren't as large in funding as a typical NIH grant or DoD grant, but they really provide the seed funding to allow us to generate preliminary data so that we can go get some of these larger grants. Like the mainstay research grant at NIH is called an R01 mechanism. It's typically $2 to $3 million over 5 years. And so these philanthropic organizations, even the smaller local ones that maybe get 50 or 100,000, we can then take that and turn that into data that allows us to get millions of dollars. And so that's another super critical funding mechanism that makes it all happen.
Yeah, Steph, can I add to that
Yeah,
too?
please do.
So I totally agree with what Brandon was saying, especially in the last part about the local philanthropic. I mean, down here in Moore County, we have philanthropic donations coming from Hormel Foundation, but we also have the Paint the Town Pink event where the whole town comes together and we do these things. Like I jumped into the ice for a plunge last year. I'll do it again this year as well. But we all come together, ice fishing challenges, everything through the month of February to raise funds to tackle cancer sooner. And it goes to these high risk, high reward funds. Now, it's usually in the ballpark of like 50 to 100,000, but it is the springboard to get these larger funds to get that initial data you need to make those new discoveries forward. So yeah, local philanthropics have a huge impact. Every dollar matters and counts.
Fantastic. And what about like pharmaceutical companies or industry sponsors? Is that anything that you want to mention that's funded your work?
Yeah, definitely. So we've collaborated with companies and we've had what are called sponsored research agreements. It's sort of like a grant where they're putting money into a specific project, but typically then any intellectual property that's generated from that then is owned by that company that's putting that funding in place to do that research. It's also kind of interesting that you bring this up because one fact that most people don't know, or people think, big pharma's doing all the heavy lifting and making all these discoveries. The fact of the matter is that 95% of all new therapeutics and discoveries that end up into products are first discovered in academia. We're literally doing the legwork, and big pharma is swooping in and making the trillions. So that's just how reality is.
Thanks for sharing that too. And if you are listening and interested in more information, like I said, we will send information in the follow-up email. Sarah, a question for you. I'm sure there's probably a lot of people on here that have friends and family that are in cancer treatment, maybe that have gone through a couple lines of cancer treatment and are looking for something in the clinical trial space. I know you mentioned that organization, the website. How else do you recommend that people advocate for their loved ones to get into these clinical trials, or how do you choose? Is it you? Is it the doctor? Is it the researchers? Can you talk a little bit more about that process?
Yeah, for sure. Most of the clinical trials um, that are for, you know, when you get sicker and sicker, and this they call it the standard of care. So there's all of the kind of the chemo, the first line, the second line, the third line of different therapies to treat your cancer. Um, if you've progressed through those, they kind of get less and less effective, unfortunately. Um, Overall, I mean, some people can have an awesome response to their fifth line of therapy or something, but overall, we see that usually around your third line or fourth line of therapy, you should probably start considering clinical trials. Um, Hopefully you never need them, but this way you can kind of get a jump on looking, because unfortunately it is quite a bit of legwork. I mean, I find that people have really told me, I don't even know where to start. And so a good place to start is with your oncologist, and talking to your oncologist about, is now a good time to start looking for clinical trials? And if so, how do I start? And they'll probably refer you to clinicaltrials.gov, which again it's just a really huge system. It's a huge database. And um, I will say that one thing you're going to need is you'll probably need to, if you do go to that website, you'll have to know your lines of treatment, what you've gotten so far. Um, if you have metastases, if you're unsure, or you go and you don't know some of these answers to the questions that the clinicaltrials.gov website is asking, you can always ask your doctor or the nurse coordinator, even for that doctor, if they have like a printout or something, genetic variations like KRAS mutation or HER2 mutation. Those can be really specific for different clinical trials as well. So you'll kind of want to know if you have any you know, it's pretty normal these days to get some genome sequencing done early in your cancer. Usually when if you have a biopsy done, they'll test, say when you're like very first diagnosed with cancer, they'll test that biopsy to make sure that it is cancer, and then they'll do some just really basic kind of genomic testing on it. So anyway, you've got a lot of info already in your chart, and that's going to be really helpful for you in your clinical trial search. And then yeah, otherwise I would start with some of those. I really, really like these AI companies that do this. There are, most of them have, it's free, and a lot of them have a nurse that will actually do some of that legwork for you. You do have to make an account, and again, it's free, but you do have to make an account, and then they'll contact you by email, and then you can kind of contact them and say, yes, I actually do need more help. And I would just take it, like, because they can call some of these different academic centers, because, can you travel? Is traveling not an option for you? Different things like that. They can help with that type of stuff too.
And then once people find you, how do you decide who is a participant in the trial?
Yeah. So the each protocol, like that guidebook I was talking about, they have a specific inclusion and exclusion criteria, and it's very nitpicky. And especially for phase 1 clinical research, which is early development, that's kind of what we do in my department called uh, uh, a developmental therapeutics clinic. We do specifically phase 1 clinical research. And so that is, the earlier stuff that is, you won't be getting a placebo. You will really get the treatment. But another thing to know is that you can self-refer to any academic center. The academic centers are usually where they'll have some of these more like a phase 1 clinical trials. So, Madison, Mayo, Chicago, and us, and, things like that, they will have more of these options. And then, yeah, as far as my, I work with multiple doctors to kind of decide. I have a wait list, actually, unfortunately, of about 80 people that are all just ready and waiting for clinical trials. And the sad part of it is there's just not enough slots. When you're thinking about cellular therapy and things like that, you can imagine, and as Brandon said, this stuff takes a while. Um, you know, they maybe can only do 5, 6, 7 of these a year. So you can imagine a lot of people just kind of waiting and kind of hoping to be one of the ones that is chosen. So it is really tough. So that's why I say if you start early and really broaden your search to try and get to a few different academic health centers, I would recommend it. But yeah, it's ultimately that inclusion exclusion criteria, which is also that's actually listed on the clinicaltrials.gov. If you click on a little button, it'll take you to that inclusion and exclusion criteria.
That's great information. Thank you. And we'll send in our follow-up email. The University of Minnesota has resources as well, studyfinder.umn.edu and also cancer.umn.edu/MNCCTN. So we'll send that in the follow-up email as well. Question for Eric. Eric, someone mentioned clear cancer cells. And I'm wondering if you can just speak to the cancer cells that you have found that are more likely to spread or maybe more likely to metastasize. What information can you provide on that?
Yeah, I can go. I'll keep it a little high level, but so not all tumor cells are the same. There are hierarchies in our tumors. Just like our tissues, we have tumor cells that have more stem cell-like function, and we have tumor cells that are more what we call differentiated. So oftentimes, the really what we can correlate with are the most aggressive tumors and the ones that spread the best have more stem-like features, or we call it generative capacity, but the ability to create more daughter tumor cells. They go into a state that we call plasticity. That means they're highly adaptable. So the tumor cells that are the best at this have a ground state or a state of plasticity, highly adaptable, and they can propagate and spread very quickly. They're also very hard to treat in those contexts unless they have something that makes them visible, like a high mutagenic load, like Brandon was mentioning, in like melanoma. But in other cases, some of them spread quickly because they have this state of adaptability that's really high. And that involves things like energy consumption, the ability to quickly change their profile, the armor that they have on with other immune cells to hide. It changes. So, and that's true not just of clear cells, but it could be true of any really type of tumor, because there's different subtypes of tumors within each. Like there's different types of gastric tumors, there's different types of breast cancers. Even within that, there's differences in that hierarchy. So it gets complex pretty fast. But a lot of the common themes are the ability of these cells to adapt and being in a stem cell-like state.
Great, thank you. Brandon, I know Terry mentioned, and I kind of felt the same way, that 95%, that number that you said in your last answer, of the pharmaceutical research comes out of academia. I think people are a little shocked at that, and that the pharma companies are making all the money. There was a question about, does the medical school get to recoup any of that money, or how does the IP transfer from the medical school to pharma, and how does that relationship work?
Yeah, that's a good question. So typically, if we're doing, funded research from NIH or DoD, that intellectual property is owned by the University of Minnesota. They file patents, they prosecute them, they pay for them, and then big pharma will come in and see those patents and they might be interested, and then they can take a license to those patents where they would give some potentially upfront fee with milestones on the back end for clinical trial deployment, different phases, number of patients treated, things of that nature. But a lot of times too, they'll take ideas that have expired. So patents only last 20 years. So sometimes they just wait for patents to expire or kind of work around existing patents. But definitely sometimes they are actually licensed to big pharma, and then funding does flow into the University of Minnesota. And different institutes or universities have different benchmarks for the amount of funding they need to get for pieces of IP. But yeah, that's the general structure.
Great, thank you. Sarah, there was a question about nursing. We have some people in the chat that are interested in your experience treating kind of the older models of cancer patients, chemotherapies, radiation versus kind of these newer cellular therapies. How has your experience changed as the treatments have kind of evolved over the last 10, 15 years?
Yeah, that's a great question. It has, uh, it's it's been pretty wild actually. You think of something, when we used to have really um terrible, terrible side effects that people who were transplanted, who got bone marrow transplants and things like that. Oh my gosh, they used to get something called graft versus host disease, where their transplant would react to a part of it would kind of attack their skin kind of by accident, or a kidney transplant would reject the body, or um, just these really terrible rejection and uh, graft uh, graft versus host. Even the chemotherapies, just terrible, terrible mucositis, and people would just get, the radiation would be, they'd get total body radiation, and they'd get like really bad sunburn kind of types of things. So it was pretty brutal. And um, now with so many, when I was talking about the whole idea of that hippo with the birds and just attacking the birds, one of the advantages of doing something like that is that you do hopefully have a lot fewer side effects when you're targeting just one specific um, piece. Um, you can really have some people can be on immunotherapies and really not notice a ton of anything. And so that's really different um, and great. And I just think it's gotten so much better. I mean, even we're doing autologous uh, stem cell uh, CAR Ts, different things like that in the outpatient setting now. You can actually get that done at the infusion center, at the clinic. I mean, the what's really exciting is the idea of being able to bring these types of innovation to rural communities and to even places that are an hour or two away. I mean, if somebody could go to Maple Grove and get some of like a CAR T. I mean, can you imagine? That would be really pretty incredible. And so it's really opened up, um, this whole scope of possibilities. But I do know that um, on the other hand, we're seeing cancers that are affecting people younger and younger. Um, people with colorectal cancer who are 31 years old and things like that. That I have to say has been a real downside, something that I have really struggled with a little more, seeing these such young people that are affected. On that CRISPR study, I think my oldest patient was 62. Almost all of them were between, 40, 50 years old. So it was just a really scary thing to have to think about. And what I think about when I say that is just your stages of grieving. The way that you handle that kind of news when you're 30 as opposed to 65, 70, 75, 80 is very different. And it takes a whole different level of care. It takes a whole different mindset. So I do think that's a big deal too.
Thank you so much for sharing. And
Yeah.
thank you to all of our panelists today for sharing your insights into reimagining the cancer treatments happening right here at the University of Minnesota. I'd also like to thank all of you, our participants, our lifelong learners, our enthusiastic question answerers. Thank you so much for coming and joining us. And since you've made it this far in the presentation, please note there will be a link in our follow-up email, and one lucky winner will win some fabulous merchandise from our store. So please fill it out and we'll send that to you. We also hope to see you November 13th for our TIL therapy fundraiser. And with that, I'll say good evening and everybody have a safe and happy weekend. Thank you.